We are based across 2 campuses, and I use the words holistic and integrated drug discovery very intentionally, because drug discovery is very much a interdisciplinary science. So we've got our medicinal chemistry platform, which our MedChem team designs and makes compounds, and they interface with the computer-aided drug discovery team, so that's more sort of protein docking work, and then more recently the AI work, which I'll be speaking about in this presentation. We also have our drug metabolism and pharmacokinetics platform, both in vitro and in vivo, and various biology teams to understand the disease biology for various pathogens and to do our screening work, including our enzymology platform for if we've got target-based programs. Some highlights of what we've been able to achieve over the last 12 years or 13 years. We've got active programs in 4 disease areas, so malaria, TB, antimicrobial resistance, and a little bit of COVID work. We've had 2 candidates make it to the preclinical phase and 1 candidate make it up to phase 2 clinical trials. Perhaps if you're not very familiar with the drug discovery approach, that might not seem that impressive, but when you consider the attrition rates involved and inherently involved in the drug discovery process, you have to be prepared for failure, and a lot of failure. We have a saying in our team where if you're going to fail and you're expecting to fail a lot of the time, you need to fail fast so that you fail cheap, which is how the whole AI will start playing into this. But just to highlight, so this was a study done across the big pharma industry for small molecule drug discovery. And for, I can't indicate, but for every drug that actually gets approved by the FDA, you maybe have 5 or so that get actually 2 clinical trials, and that all started with thousands or tens of thousands of compounds even entering the drug discovery pipeline. And that's before you've had to screen the millions of compounds to even find those hits. So you really are in for a difficult challenge when you start getting into the business of drug discovery. And never mind the expenditure involved, so the average cost is sort of estimated between $1 and $3 billion and 10 to 15 years of development to actually get 1 single drug approved. We've seen the same attrition rates in our history, so the number of programs you can see drop off as you go through the different advances in the drug discovery pipeline, going from about 65, 64 programs entering the pipeline down to just 5 that make it to the selection phase going into preclinical trials or preclinical profiling. So how we try and address this in the drug discovery process is by using these things called screening cascades, which is where we're trying to triage our candidates before we progress them further and further in the pipeline, so that we're only progressing the most promising compounds further in the pipeline. And generally, these are also structured both in terms of time intensity and expenditure for the different assays that you're trying to conduct. So it's relatively cheap to do an enzymology assay, but once you start getting to in vivo PK, that starts running into quite severe expenditure. So as our first foray into the AI world, we have about 12 years of data in-house that we could leverage from these various assays. And we partnered with a non-profit entity from the UK called the Acilia Open Source Initiative. And they came and did a research visit with us to both develop tools, but also impart skills which we didn't have until that point. The plan was to develop machine learning models corresponding to each of our assays so that we could have a virtual screening cascade aside to the actual screening cascade so that we could pre-prioritize compounds to go into the experimental pipeline so that we're trying to reduce the attrition rates that we'll see and the resource expenditure associated. As you'll see in a bit, I'll describe that this sort of evolved into a new direction, which was more than just developing some machine learning models once off that wouldn't be incorporating new data. We extended this to develop a whole machine learning pipeline, model training pipeline that could automate some of the process of finding good models and allowing people with limited data science skills to also implement those models, thinking particularly about a lot of the researchers we support in other African continents where they don't have such a big ecosystem to draw on, where there may be just one lab chemist, but also interested in implementing AI tools to their research. So just to go into a little bit of technical detail to maybe make it a little bit more tangible, one of the problems we face, in addition to selecting which machine learning algorithm we're actually going to be using, is also how do you describe your compounds in a way that the computer can actually leverage it. And some examples, perhaps you featureize just the presence or absence of different chemical moieties in your group or the number of them. Perhaps you do sort of more abstract biochemical properties, so number of rings or hydrogen bond acceptors and so on. Of course, this doesn't really cater for the three-dimensional aspects of a molecule. It's just indicating very small local regions. So one could also think about some all-to-all matrix, say electrostatic force, some sort of three-dimensional property from all atoms to all atoms. Compounds are naturally represented by something called a SMILE string, which is just a piece of language text, so you can apply different language processing models to this. Compounds are obviously naturally represented as graphs, so vertices as the atoms and edges as the bonds, and you can apply then the whole field of graph neural networks and graph theory to that. Up to even more exotic fingerprints, which is where you convert these compounds into pixels of different properties, and that can be fed into convolutional neural networks as an image. So to automatically select which of these are best, we built a platform that lets us run through all of these to find the most descriptive and combine this. So starting with some pre-processing steps in our pipeline, we describe a molecule in a panel of different ways, 6 or 7 popular methods from literature trying to take different angles at describing the molecule. In some contexts, perhaps the three-dimensional aspect is more informative. In other contexts, perhaps the problem you're trying to solve is very specific to a very local region, the presence of a feature in a molecule. Once we've described the molecules in multiple different ways, then we pair that with many different machine learning algorithms to try and find the pairs that are most predictive for your task at hand. And then finally, use those top models to train a model of models or a meta model as your final thing to implement. All of this is happening in the background. As the data scientist or even just as a lab chemist, you only need to know 2 commands from Ubuntu command line terminal. But what we found really helpful as well is if you've got someone on site to implement those tools and help explain it to the chemists, but also see what they need and cater that into how you're designing a model and the cutoffs that you use, that's really powerful as well. So again, just to get us quickly all onto the same page before we start looking at how the models sort of work at a very high level, everything I'm speaking about here is something called a binary classification model, which is where you've got just 2 categories, and in our case, an active and an inactive. So soluble, insoluble, or toxic, non-toxic, based on a cutoff that we've defined through working with experimentalists. And one metric that you can use to measure how this model is doing is something called the ROC curve. And all you need to know is that the more area under the curve you have, the better. The closer you are up to that top left corner, the more predictive your model is. If you're at a 0.5 area, that's a random model, it's not helpful. If you're at an area of 1, that's a perfect model, very unlikely, should be red flags there, but you're trying to get as high as possible in that curve. And what we saw when we built the model on our in-house data, so for malaria specifically, which you see in the top left, we get some really good predictive performance on our actives versus inactives in our potency assay, as well as for our MTB model. And what we also found, which is really helpful, is although it would be great to always be able to absolutely classify active versus inactive, even if that wasn't the case just because of how the model has been calibrated, the model is still pretty good at ranking your top compounds first with the highest scores and your worst compounds at the bottom. And ultimately providing you with this hit enrichment, which you see in your second graph, where in the first column is the background activity rates that we've seen in our data set. Once you've ranked the compounds in the middle compound, you see all the compounds in red, all your actives are ranked much higher, and in your bottom 50 on the right column, all your inactive compounds have been ranked much lower. So when you think about this from the perspective of a medicinal chemist, if they're starting to create ideas, run this through the model, and then start making those compounds in that order, they're going to be coming across their promising compounds much more quickly than had they just been going about it in the traditional way. Then looking at this across all of our assays, so on the vertical axis is the ROC curve score. In general, all the models did pretty well. For the models that had fewer data, particularly the cytochrome P450s on the right-hand side, which is metabolism related, where we only have very limited in-house compounds, those did a lot worse, but you can think about approaching this with literature data or transfer learning approaches to remedy this. And yeah, this is just sort of an overview of the data that we have in-house and the active-inactive ratios of each data set across activity, solubility, tox, and intrinsic clearance, which is a PK parameter. We were then also interested in looking at this in a more prospective sense. So we looked at all compounds produced within 2 chemical series. So those are where you've got a core scaffold that is kept relatively constant, and the medchem team is just placing different appendages onto the side to try and optimize the properties of that compound. We wanted to see how predictive this was going forward. If in this graph, for instance, if you see a green dot, that's where the model got the prediction right, and purple is where it got it wrong. But what we were interested in asking as well is, when the model gets it wrong, is it just unsure, or is it actively pushing you in the wrong direction, which would arguably be worse than not having a model at all? In general, so the top plot is for a malaria series. It does quite well. And in the bottom plot, we see very few purple dots, so very little sort of wrong direction pushing, but more just unsure if it gets it wrong. And more importantly, what you'll see in the plots on the bottom right is we'll see actives plotted in the red graph on the right and blue on the left. And we're just trying to see that separation between the classes. And your compounds that are getting the highest scores are still your most active compounds. So even when it's not that great overall, you're still finding good compounds first before you start finding false positives. It's worth pointing out that this was actually accepted into Nature Communications last week. So if you're interested in finding out more about it, you're welcome to look up our paper. Then, of course, we also want to make sure that we're not fooling ourselves. We want to be looking at this in a prospective sense with data that we've never seen before, or the model has never seen before. So all these models were developed on data up till 2021. So then we could look at all of our 2022 data and see how well did the models do, how well could we have done had we had them on the ground running with our teams. And as you can see with the rock curves, we get some good enrichment. And with the just plotting the different compounds with the actives and inactives in the red and the blue respectively, we see some good separation between our compounds, and particularly the compounds getting the highest scores on the y-axis are the compounds that are most active, and you're going to be finding that hit enrichment before you actually progress your compounds to the experimental cascade. We could then also do some interesting data exploration, trying to find out some helpful tips for our medchem teams. So here we were interested in looking at the influence of data within a chemical series versus data from a more general chemical space. So then looking at each individual chemical series, what we did was we removed all of those compounds out of your data set and tried to predict it, see how well we do, which is the leftmost column in the middle panel. And then we slowly start adding training compounds back into the training set from that chemical series to see how quickly we improve, to see is there an ideal number, a good ballpark figure to tell our medchem team, look, we're not sure how well the model is going to do up front, but make us 10, 100, 1,000 compounds. We don't know. How much do you need to be predictive in your chemical space? So a good sort of rule of thumb that we found was between the chemical series, generally, if you can have around 30 compounds, that was a pretty good initial starting point for your teams to go and make a diverse chemical set. And then we could most likely build you a pretty predictive model going forward after that. Then exploring the second theme, which is if you have data for a chemical series, should you build a model just from that? Or should you also include external, less related data? Does it have any information to add? So what we did was we built models where we only have a training set based on a certain chemical series. So for instance, if we look at the red bar in the middle, in the far right panel, we train a model that has only seen compounds from that chemical series, and we see how it does. Then we add in our background library of compounds, which are not necessarily from that series, from that chemical series of interest, but just from a general chemical space against malaria. And do we see any improvement? And so the bars here represent of the performance I had left to achieve from my model, how much do I actually get by adding just background, less related data? And we saw that in general, it was a good idea to include all data that you have access to, even if it's not specific to that chemical series, which of course is a very helpful tip to know when you're trying to build models to simulate your chemical space. Lastly, just to finish off, then we also needed to prove that this method was adaptable to chemical series, not just in the H2D chemical library. So we applied this to the therapeutic data commons benchmarking sets of absorption, distribution, metabolism, excretion, and toxicology data sets. And we generally saw pretty good performance, near state of the art in the top 4 almost consistently. And again, if you're interested, I would encourage you to go and read our Nature Communications paper. Finally, just to thank the organizers for inviting me to speak, and our team at H3D, as well as the Acilia collaborators and our AI mentors. Cool. Thank you very much.
Thank you, Dr. Hlozek. Are there any questions? One question. 2 questions?
It's a very interesting paper. Definitely going to go read it. Based on the work that you guys did you guys at some point when going through these compounds and identifying optimal ones, separate them out into functional groups and then work back for molecule design?
So that would definitely be a next step, and that kind of touches on this interpretability of your model. Yes. Can you understand why it's making the predictions that it's making? We haven't explicitly looked at that yet, but we're kind of approaching it from a different angle where we're starting to apply these generative AI techniques, which are algorithms that are proposing new molecules, and it's getting feedback on how those are doing based on those models. And then if you can analyze those results, which we've just started doing, you can kind of get a sense of what is the model thinking is a good compound. You can sort of analyze through clustering methods and that from all the compounds that are proposed, which are the ones that seem to have a theme across. But there are also other techniques to specifically investigate feature importance and like Shapley values. And yeah, so it is the next step to do.
Hi, Jason. Sabri here from UCT as well. But I wanted to understand whether the model, I may have missed it, but does the model also train for the possibility or the ease with which you can effectively synthesize the compound as well? Because I think that's also where the proof is going to be in the pudding, is whether you can synthesize it, ease of synthesizing it, looking at conditions of basically manufacturing and scaled up manufacturing downstream as well. But then over and above that, once you can effectively build the compound, test it, you know, and then to reinforce the lab data basically with what you've done in silico.
Sure. So yeah, good question. So from this initial work, we weren't incorporating synthetic accessibility into this because our medchem teams were coming with proposed molecules, and through their expertise, we would presume that they've already identified an optimal chemical space to operate in. But for sure, for like generative methods, if you just let those run wild, you'll see some pretty exotic compounds, which maybe they work, but you would never be able to test. So you get models that do predict that synthetic accessibility, and you can build that into the scoring function to get it to move towards something that you can actually make and then test and prove the algorithm.
Hi. Tell me, how much of this is open source, or has this become a new product of H3D and new IP? Is this something that we need to take into account in skills development of biotechnologists?
So all of the tools, all of the framework is completely open source. It's on, so Acilia's mission as well is to develop data science tools for the low resource community African countries. The data itself, so we've released models with anonymized models where you don't see the actual data that they were trained on, but you can still make use of them. I think the data aspect, the raw data that you're building from, that has a lot more IP constraints, and that's a bigger issue to deal with. But all the code and the building the models, if you come with the data set, all you have to do is install this from GitHub, and it will, with 2 commands, you can run your models and develop it.
More question for the sake of time?
Perhaps just zooming out a little bit from a non-deep biotech perspective and just trying to understand the evolution of technology and the speed with which it's moving, the extent to which generative AI has impacted your work would be interesting for me to understand just as a layman, and when did that start having an exponential effect on what you're doing?
So these models were officially sort of implemented maybe 3-4 months ago. with the generative models only starting in the last month or so. So I think it's hard to say impact yet, but certainly very interesting having explored the aspect if you incorporate synthetic accessibility or not, and you just see actual feasible results versus wonky things. I think that'll be a very interesting follow-up work in the next few months to go to the MedChem team and say, what looks appealing to you from this set? Can you make them and test them? Let's see how we do, and can we iterate towards a good workflow from that?
Right, thank you, Dr. Hlozek.